scientists have created a functioning mouse embryo in the laboratory by using stem cells in a world-first scientific breakthrough. The researchers created the artificial embryo using two types of stem cells, the body's master cells and a 3D scaffold on which they can grow. The discovery is hoped to shed light on the development of life in its earliest stages and could explain why 2 out of 3 miscarriages occur during early pregnancy. Making an embryo naturally would use sperm and egg cells implanted in a womb. Stem cells can make any cell in the human body, but making them grow into complex three-dimensional structures like an embryo or human organs in a laboratory has foiled scientific efforts to date. Embryonic stem cells do not turn into the cylinder shape of an embryo on their own, but need a helping hand from other cells which would be present in the body but not in the laboratory. To help them along, the scientists use 2 different kinds of stem cells, embryonic stem cells and another kind of stem cell which acted as a 3D scaffolding to hold the developing embryo in place. This other kind of stem cell, called a trophoblast stem cell, goes on to create the placenta in a normal mouse embryo. The research, by Sarah Harrison and Magdalena Zernicka-Gitz at the University of Cambridge, was hailed as a masterpiece yesterday by fellow scientists. Professor Zernicka-Gitz said the key to the breakthrough is the extra embryonic cells that acted as a scaffold. She said both the embryonic and extra embryonic cells start to talk to each other and become organized into a structure that looks like and behaves like an embryo. It has anatomically correct regions that develop in the right place and at the right time. The 2 different types of stem cells communicate, telling each other where to go, the author said. She added, "We knew that interactions between the different types of stem cell are important for development, but the striking thing that our new work illustrates is that this is a real partnership. These cells truly got each other. Without this partnership, the correct development of the shape and form and the timely activity of key biological mechanisms doesn't take place properly." The artificial embryo followed the same pattern of development as a normal embryo and was 92% similar to a normal mouse embryo. The embryo is unlikely to develop into a normal mouse if implanted in a womb, as a further kind of stem cell would be needed which would create a structure called a yolk sac which would provide nourishment to the growing bundle of cells. Professor Zernicka-Gitz said this development could help human embryo research by overcoming a major barrier, a shortage of embryos. Currently, embryos are developed from eggs donated through IVF clinics. She said, "We think that it will be possible to mimic a lot of the developmental events occurring before 14 days using human embryonic and extra embryonic stem cells using a similar approach to our technique using mouse stem cells. We are very optimistic that this will allow us to study key events of this critical stage of human development without actually having to work on embryos. Knowing how development normally occurs will allow us to understand why it so often goes wrong." Dr. Andrew Chisholm, head of cellular and developmental science at Wellcome Trust, said, "Professor Zernicka-Gitz's work really shows the importance of basic research in helping us to solve difficult problems for which we don't have enough evidence for yet. In theory, similar approaches could one day be used to explore early human development, shedding light on the role of the maternal environment in birth defects and health." Professor Zernicka-Gitz recently pioneered a technique to develop human embryos to 13 days after fertilization, but calls by her and other scientists for the legal limit of 14 days to be extended have been dismissed by the Human Fertility and Embryology Authority. Dr. Dusko Illich, reader in Stem Cell Science, King's College London, said of the research, "Another masterpiece of recreating in vitro in a laboratory the earliest steps of embryo development coming from the Zernicka-Gitz lab. A beautifully conceived and executed study demonstrating interplay of different cells in different cellular compartments within the first days of mouse development." Professor James Apche, chair of Stem Cell Research and Regenerative Medicine, Heinrich Heine University, said the research meant it will now be possible to better study and understand why certain gene mutations affect normal embryo development.